So without further ado, we should start.
Thanks, everyone. We're going to get started with session 2.
Hello.
Hello.
Hi, everyone. Thank you so much. I'm going to have to use my loud New Jersey voice next. It's going to get even louder. No, I'm just kidding. Thank you all so much. We're back from our bio break, and we're really excited for the second session. And I'm going to hand it over to our moderator, Lavinia.
Yeah. Thank you. And it's an honor for me to introduce Ruth. She is a professor at the University of Oxford. I met Ruth 7-8 years ago. And actually, I contacted her because she was and is a pioneer in this field. And it takes a person to change a village and a village to change the world. So she is certainly someone who is going to change the trajectory of neurodegeneration and dementia, and her work is really seminal as many other 35 years ago. Thank you, Ruth.
Thank you very much, Lavinia. And I'd like to thank the organisers also for inviting me, and say I'm sorry that I won't be there to see people in person, but I hope in future to meet them. Anyway, I'll be talking about HSV1 and Alzheimer's, and I want to start by giving a little bit of micro history. So next slide, please. Oh, what's happened to the slide? Oh, here we are. Good. Well, you might well ask, what was the idea? Why a virus and why herpes? The reason herpes is because herpes family can, like other viruses which have been mentioned already, like measles and HIV, can cause neurological disease. In other words, people seem to think of them as acute, always as if a virus flies in and then flies out again, having done its damage or killed the person. This isn't the case with quite a lot. Some of them can stay there and cause damage after a long time. Anyway, the reason for HSV1 being a likely candidate is ubiquitous. It infects nearly everybody. In countries with high socioeconomic level, it's something like maybe 60+% of the population. In ones with poor socioeconomic level, it would be much higher proportion, 80+%. Once it gets in, it resides lifelong in latent form in neurons of the PNS, the peripheral nervous system, but it can be reactivated often. And in some people, this causes cold sores, herpes labialis. And the point is, it's in some people. In other words, many people can be infected, but not everybody is affected. And that's an important point, which a lot of people don't seem to understand. Anyway, the reactivation can occur through stress and various other forms, immunosuppression, UV light, menstruation, and so on. In a very rare acute brain disease, HSV1 encephalitis, importantly, the regions affected are those most affected in Alzheimer's disease. And it was suggested by Mel Ball in 1984, I think, that this might make a connection between the two. Next slide, please. Next slide. Thank you. Well, we decided in 1989 to have a look at brain samples by what was the then new method of PCR, which was a bit of a nightmare because getting it to work was difficult, and there was also extreme troubles with contamination, which we took enormous care to try to avoid or prevent. And so we used look for a viral sequence, viral thymidine kinase sequence by PCR. There had been some previous work by using much less sensitive techniques like in situ hybridization, but they really weren't adequate, and they weren't consistent either, which isn't surprising because if you're looking for a needle in the haystack, not many methods will detect that needle. Anyway, what we got was in a way disappointing. We were very naively hoping that we'd find the virus present in a high proportion of Alzheimer patients and a low proportion of new elderly normals, age-matched normals. What we found was there was slightly more. The frequency, which is the y-axis here, frequency or prevalence was fairly similar. And what this was suggesting, rather like the cold sore situations, a lot of people get infected, but not all of them by any means are affected. However, I don't know what's happened to the y-axis. Sorry, this is elderly. This is Alzheimer's, elderly normals, and this is these are young people of whom we had rather few. N is the number of brains, but in fact we use specimens from various regions of the brain. I don't know what's happened to the x-axis. It's disappeared. Perhaps the slide could be, I don't know, perhaps it's possible to show. There should be here labeling what, saying what they are. Next slide, please. Well, we thought about it, what could this be due to? And one obvious thing that came into account was the possibility that there was a difference in genetic factor between those who developed Alzheimer's and those who did not, even if they had HSV1 in brain, even if it was a factor. So the obvious genetic factor to look at in the early 1990s, which was, by the way, the time we got the paper published, which we took extreme battles to get it published. It took detection of the virus in brain, took, I think, probably well over a year and a half, but eventually we did get it published. Anyway, we decided to look for type 4 allele of ApoE because that had come into prominence in the 1990s as a major susceptibility factor for Alzheimer's. So we looked at that in our brain specimens, and what we found was that there was a very high ApoE4 allele frequency in people. This, again, the labeling's vanished, I'm sorry. This is, the first column is labeling HSV1 positive in brain Alzheimer's disease patients, and this is HSV negative brain. And you can see this is a very high frequency here of ApoE4. In the elderly adults, again, sorry, the labeling's vanished, the values of HSV, of ApoE4 frequency were low. So there's a strong association of ApoE4 with HSV1 present in brain of Alzheimer's disease patients. Association doesn't prove causality, of course, but we were able to argue against its being artifactual, like people with an ApoE4 allele being more susceptible to infection, and that sort of argument which we could discount. Next slide, please. Well, having found that, we then thought, what possible linkages are there between HSV1 and the particular features of Alzheimer's disease brain? And so we looked at infected human neuronal cells, glioblastoma in this case, and we also were able to get from a lab that was doing infections of HSV1 infected mice, we were able to get specimens of brain. And these were labeled for A beta. The top sections are all infected, the bottom ones are uninfected. And as you can see, A beta, nice staining in the infected cells for A beta, and the same in the infected mouse brain, very little or nothing in the uninfected. And these are here stained for p-tau. We stained at various phosphorylation sites, and they all showed similar things. Again, uninfected, low labeling, which is, by the way, explainable, so I won't go to that now. So there was a very interesting linkage. The experiment with A beta were the first to show that the virus could cause formation of A beta on infection in both mice and in infected cells. As for the p-tau, we were probably the second to discover it. Next slide, please. Then we tried something very, very speculative, which we didn't think for a moment would work, but it seemed well worth trying. And that was to look for the location of the HSV1 in brain sections by in situ PCR, which was an extremely difficult technique. I think we were the only people in the world doing it. In fact, we were probably the only people in the world, amongst the only people in the world doing anything on herpes and Alzheimer's. But this particular technique, which could be, of course, used for a lot of other things, was very rarely used. And it took us several years to set up. Really very difficult, but very rewarding as it turned out. So we were really delighted when we found very specifically co-localization. The first one shows one section, which shows you here the HSV1 DNA stained, detected by in situ PCR. and then shows you a plaque stained by thioflavin S, and then the merger, which, as you can see, the two completely coincide. And then below, there's another section in which the A beta was stained by immunochemistry, sorry. And what we found was about 72% of the DNA is present in AD plaques, but only 24% in the brain aged plaques in aged normals. But nearly all the plaques contained viral DNA. So the next slide shows, please, next slide, shows what happens when you, we decided we'd try to find out if acyclovir, which is known to affect HSV1 synthesis, it interferes with the viral synthesis, viral DNA synthesis. So we decided to try that on infected cells and see how it affects the levels of A beta and P tau. As a sort of guide, they were used as sort of signals to show what happened on using an antiviral. And what we found was quite striking. Top shows the concentration, the level of viral proteins, which goes down considerably, but doesn't go to zero. There are perfectly good reasons for this, but I haven't got the time to explain it now. Similar thing happened with A beta, which goes down quite a lot, but does not go to zero. And this one, I'm sorry if you can see it, the level goes down. This is P tau, and in this case, the level went down to just about zero. The bottom axis is the concentration of acyclovir, well, as you can see here. And it probably reflects the time at the viral sequence when the proteins are made. Next slide, please. So this is just to summarize the earlier work. If you're anything like me, you probably forget what's been shown already. So perhaps it needs a prompt. At least I find I need prompts. So what we found was that the virus HSV1 DNA resides, and we found it was latent, but reactivates during, we didn't find this, but this is known, reactivates during stress in the trigeminal ganglia. We assume that the same happened probably in the brain of many elderly people. And the reactivation damage is presumably limited and localized. In other words, what you might call a mild HSE, herpes simplex encephalitis. And I say that because if it were obvious encephalitis, the people would have very different symptoms. They'd be seriously affected, very, very damaged, and either killed or these days, given antivirals, they might survive severe memory damage. So obviously they weren't, you couldn't call it a wholesale or a large scale reactivation. But as we're saying, the virus DNA there is probably at a very low level anyway. And then what we found after that, a few years later, again, awful trouble getting this published, but eventually got published in the Lancet after a great deal of argument. This is it. I should say, in agreement with other people, that getting funding for this work was incredibly difficult. And we survived on really a shoestring. And the money came not from the major grant-giving bodies by any means, but from several small charities who were daring enough to try a new approach or to support a new approach, which was very good because otherwise it would have collapsed altogether. And other people have come into the field in the last 10 years because funding has become available, but it certainly wasn't in those days. And we were almost the only people doing it for a large number of years. Anyway, so what we found was with ApoE4 that HSV1 in brain and ApoE4 confer a strong risk of AD. I didn't show the graph of this, but it's being quite striking. ApoE4, we discovered, is a risk also for cold sores, which was really very nice and very significant, suggesting that it's a risk in both the CNS and the PNS. And HSV1 infection of human brain cells and mouse brain causes AD-like changes. and the viral DNA is located within amyloid plaques, which suggested that it was encaged by them, but we didn't dwell on this because we didn't have any further evidence, but the co-localization was very striking. Then we found that antiviral treatment, stopping viral DNA synthesis, reduces greatly A beta and P tau production. What I haven't mentioned, no time, was that we tried some vaccination experiments, unfortunately rather limited because the person we were collaborating with retired, but we found that mice vaccinated against HCFC1 were protected from the establishment of viral latency in brain, which was really very encouraging, and I wish we could have continued it, but didn't. And in a completely different set of experiments, we discovered that ApoE determines the severity of damage or the susceptibility to infection by diverse pathogens. And interestingly, type 4 is not always the bad guy. We found it strongly protective in the case of hepatitis C-induced liver damage. The reason we chose to look at that and also a number of other viruses, we selected them on the basis of the criterion that the virus enters the cells through receptors, which were also receptors for ApoE protein, the different ApoE proteins. So we thought that it may be that some of the one or two of the isoforms are more easily bound or more easily dislodged by comparison, by contest with a virus. So in other words, it would determine the degree of infection and therefore the degree of damage. I should say, by the way, about the discovery of an infectious pathogen, which really was the first time. It was the first definitive discovery of an infectious pathogen in supposedly sterile tissue, the presence of viral DNA in brain. We did the work in about early 1990, but it didn't get published till 1991. Next slide, please. So as a result of all this, we formulated the HSV1-AD concept. The virus resided latently in brain of many elderly people, but can reactivate and probably repeatedly. It then causes damage, including inflammation, which is limited and localized, but cumulative, leading to eventually to AD. HSV1 probably travels from the trigeminal ganglia to the brain in late middle age on decline of the immune system, or perhaps enters the brain via the olfactory nerve. The virus in brain, together with the genetic factor ApoE4, confers a strong risk of AD. And probably there's more damage in ApoE4 carriers or possibly less repair, we don't know. And then ApoE4 is a risk for herpes labialis, in other words, for damage in PNS as well as CNS. Antivirals protect against the HSV1 damage in cell cultures. And then now well over 500 studies using very diverse approaches, epidemiological, molecular, and cell biological, virological, and GOS methods, which support this. There are just a handful, a few, which do not agree. And in most cases, I found that you can explain it quite easily by people, for example, including controls who are actually infected but asymptomatic. They're included as controls, although of course they're not really controls, that sort of thing, or else people haven't quoted the sensitivity of their techniques. And if you, it's quite possible that they're using far too inadequate sensitivity to detect what's almost certainly present, the virus in the brain at a very low level. Next slide, please. So from all this, there seem to be two main questions arising. I mean, there's another question as well, which I'd love to solve, but I'm sure other people will be working on that. And I would like to, unfortunately, it didn't work out, a collaboration that was supposed to look into that. Is that, and that is the question of whether it's one agent, one infectious pathogen like HSV1 acting with others, or do some others act together? Are there several agents which act together, in other words? I mean, the main herpes virus and also the main bacteria being implicated. like chlamydia pneumonia and Borrelia and the oral bacteria. Anyway, these questions that we tried to answer, well, not that one, the ones we've had some success in looking at, I think. Is there evidence of HSV1 reactivation in brain? And if so, what causes it? And secondly, is there evidence that the virus reactivations lead to AD? Next slide, please. Well, what causes HSV1 reactivation? And there are many types of injury now known. Well, in the last few years, it's been shown that infections, as somebody mentioned today, infections cumulatively probably, but including COVID-19, and also inflammation, hypothermia, immunosuppression, and also axonal damage, which can happen during neurosurgery inadvertently or various other ways, including head injury, cause HSV1 reactivation. But I'll be showing the head injury later on because we're quite excited about that, as we found two main causes, infection and head injury, two main risk factors for Alzheimer's disease, well, I should say, have apparently work through HSV1 reactivation. Next slide. The evidence of HSV1 reactivation in living brain. Well, we found that HSV antibodies are present in CSF of AD patients and age-matched normals. In other words, it suggests that HSV1 replicates and is expressed in brain. And we looked for them because after HSE, which is, as I said, very different, being much, much more severe, vastly more than we think is happening here. But after HSE, these antibodies are present for quite a long time after the incident. So we thought they might well be present in CSF, and that's where we found a reasonably high proportion. Anyway, there have been other studies on this for quite different reasons. Routine labs doing routine studies on HSV, on CSF. And in two labs, two studies, separate studies, HSV1 has been detected in the CSF of far more people than would be expected after the very rare cases of HSE, which usually the most obvious ones are something like a few, 3 or 4 cases per million people. So you can see it's really very rare. But the virus is detectable in CSF of far more people than you'd expect after that, just after HSE. And that suggests that limited virus reactivation is relatively frequent. This is what these authors suggest. And there have been many cases of so-called mild or atypical HSE and of recurrent HSE, but just case reports individuals. Then work I was involved in way back in 1986, we looked at patients who've been immunosuppressed by in situ hybridization, one of these much less sensitive techniques, but really it was very informative. And at the time, we didn't really, I think, stress it as much as it could have been. I don't think the paper's at all well known, but we looked at HSV1, if we could detect it in brains of people. We couldn't find it in normal elderly brains or normal brains, probably because the sensitivity was far too little. But in those who were seropositive for HSV and who were immunosuppressed, it was present. In those who were not immunosuppressed, it wasn't present, even if they were seropositive, as I said. And those who were seronegative, it wasn't detectable. So presumably, it looked as if what was happening was the immunosuppression caused reactivation of the virus, which made it detectable by this rather insensitive method. And I should say, in affected mice, reactivation is far more efficient than in immunodeficient than in normal mice. I'm sorry, the bottom of all these slides is missing. I don't know what's happened. They weren't in my version. I don't know what's happened meanwhile. Anyway, next slide, please. This is just showing you the immunosuppression results, which I won't go through to without taking too long, but it does show very clearly the detection in brain from people who are immunosuppressed and HSV seronegative. Next slide, please. Well, as I mentioned earlier, infections are now known to be a major risk factor. for AD stroke dementia. I'm using those terms interchangeably because in a lot of cases, when you look epidemiologically at these things, very often people are marked as having dementia but without any specification as to what type it was. So have to use that term. And also some vaccines is well known now and very exciting, I think, work that some vaccines decrease the risk of Alzheimer's. And in fact, the first paper showing that for shingles came out in, I was involved in that work with Lofat Anand in 2021, along with work by Scherer et al. And since then, there have been about 3 or 4 other studies, 4, I think, actually, all of which have shown increased risk of AD, so decreased risk after vaccines, either Zostavax or, well, was it either Zostavax or Shingrix. So that was really quite exciting. And anyway, so cumulatively, the infections increase the risk, although we found that shingles caused only a very slight increase. Zostavax and Shingrix reduce the risk of dementia. And we suggested way back in 2002 that we asked if the increased risk, we published this in answer to a paper by Vero et al., which showed that vaccines, certain number of vaccines did cause increased risk of Alzheimer's or dementia. And so we wrote to suggest that is the increased risk a result of reactivation of latent DNA in brain. I'll try, yes, sorry, in that case, I'm going to have to go to the summary, sorry. Is it caused by reactivation of a latent HSV1 in brain, and do vaccines reduce that risk? Could you show the next slide, but I might want to move on from that. Is what we've been doing is looking at 3D cultures where you can make, when infected with HSV1, this is work by Dana Cairns et al., and they found that HSV1 infection induces AD-like phenotypes. This was published in 2020. You should easily be able to find it. They found that it produced A-beta, p-tau, and also other various cytokines and so on. I think if we just skip the next 2 slides, which are pictures, next slide, please, which shows that if you infect with it, yes, thank you. If you infect with VZV or HHV6 and certain other agents, you can reactivate quiescent HSV1 latency it can be induced in these 3D cultures by treatment with acyclovir. And if you use these viruses or other pathogens, you can get reactivation, and then you get again the appearance of these AD-like phenotypes. So the next slide shows a summary of these results. I'm sorry, I haven't got time to show the pictures, obviously. So this is a summary. HSV1 infection causes AD-like phenotypes, accumulation of A-beta, p-tau, gliosis, and inflammation. VZV and HSV6 do not cause accumulation of A-beta and p-tau, suggest they're not direct causes of the disease, but they produce gliosis and neuroinflammation. We think it's a neuroinflammation that's causing the reactivation. And VZV and HHV6 added to quiescently infected cells reactivate latent virus, inducing AD-like phenotypes. And very recently, we found that traumatic brain injury causes an accumulation of A-beta and tau, gliosis, and neuroinflammation. And it causes, importantly, reactivation of latent HSV1, inducing AD-like phenotypes. And these data are consistent with infections and injury causing HSV1 activation in brain, thence AD, and with vaccines, so for example, against shingles, reducing the AD dementia risk. Next slide, please. Well, I don't know if I'm going to have time to talk about this, but I've talked about the vaccines and the infection. And this is results of other people showing AD risk is greater in HSV1 infected people than uninfected, and antivirals decrease the risk. And cognitive decline is greater in ApoE4 HSV1 infected humans. There's genetic support for a viral role. These support a causal role for HSV1 and AD, not just an association. Next slide, please. It's getting to the end now. And is there evidence of HSV1 involvement in AD? Repeated reactivation of HSV1 in brain of infected mice causes AD-like phenotype and cognitive decline, and synaptic function is reduced. This is done by the Rome group, Grassi and Palomara, who've done a lot of experiments on HSV1 infected mice, which I think very, very worthwhile. And then the work that we've done on 3D cell culture leading to AD-like phenotype, so showing experimental evidence, not directly in brain, but in animals and in our 3D culture. Next slide, please. So conclusions. Answer to the two questions. Is there evidence of reactivation in brain of HS1 leading to AD? Yes. In models, infection and reactivation in 3D cell culture and in infected mice leads to AD phenotypes. In live infections, other injuries are known to reactivate viruses. Also, HSV1 affected people are at higher risk than unaffected. This suggests that HSV1 in brain is pivotal in increasing the risk of AD. We therefore propose that after brain injury, including infections, it is HS reactivation that leads to the development of AD dementia. In other words, the virus is literally a major cause of the disease. Thank you for listening. Apologies if I've taken too much time. Thank you.
Thank you. Unfortunately, we cannot take any question. I will ask you the question on the chat and maybe you can reply. We have to move on to the next speaker. Thank you so much, Ruth. It was a pleasure having you today.
Thank you.
I would like to introduce Dr. Elizabeth Breitschwerdt from Columbia University. She's an expert in the genetic susceptibility and the innate immunity genes that really predispose us to infectious linked neurodegeneration. Thank you.
Sorry, Dr. Bradshaw couldn't be here today, but we have Zena.
Okay.
No worries at all. She's here with us also from Columbia, and she is going to be presenting their work. And we really look forward to it. Thank you, Zena.
Yeah, I couldn't look at that picture.
All right. Well, thank you so much, Nikki, for organizing this wonderful day of science. As Nikki said, my name is Zena. I recently defended my PhD with Betsy Bradshaw at Columbia. And today I'm really happy to be sharing with you the work that we're doing to understand how HSV1 affects microglia and the role of AD genetic risk factors in kind of mediating that interaction. So we've just heard a lot about the association between HSV1 and Alzheimer's disease. And right now I'd like to take a little bit of a deeper dive into that association in microglia. So AD genetics really point to a central role for microglia in disease pathogenesis. GWAS have identified several genetic risk factors for Alzheimer's disease that are either exclusively expressed or highly enriched within the CNS in microglia. And when we look at the genetics to try to understand, is there a shared pathway that these converge on in microglia? Is there something that we can kind of glean about the functional implications of these risk factors in microglia? We actually see a very interesting trend such that for the genetic risk factors associated with microglia that we know their direction of effect, they all converge on decreased microglial activation, such that there's either increased expression or gain of function of inhibitory molecules such as CD33 or PILA, or there's a loss of function or decreased expression of activating molecules such as PLC gamma 2 or TREND1. And this leads to an overall phenotype of decreased microglial activation in Alzheimer's disease. And for anyone who wants to take a deeper dive on the subject, we put out a review article going through each of these genes a couple years ago. Now, a very important function of microglia in the brain is the immune response. And here the genetics are pointing us to a dampened ability of microglia to mount an immune response associated with AD. So this led us to start to think about, what's the interaction between infection and microglia in Alzheimer's disease? And we decided to really start by focusing on HSV1, as this pathogen has been so well linked with AD, as Ruth just beautifully outlined for us. So today I'm going to talk about our work examining how HSV1 infection alters microglia function and how AD genetic risk factors kind of can influence that infection. So the questions that we initially started out trying to address are, how does HSV1 alter microglial phenotypes? And secondly, how do AD genetic risk factors influence that relationship? Now, in order to study functional genetics in human microglia, it's very challenging to get enough microglia from primary human brain tissue at the scale that you would need to tease out the impact of genetic variants. So we actually employ a monocyte-derived microglial model to study genetics from primary human cells. So we take monocytes isolated from the blood of healthy individuals, and we expose them to a cytokine cocktail that allows them to differentiate into microglia-like cells. And these cells ultimately are more similar to human microglia than mouse microglia are to human microglia. And if anyone's interested in this method, we have a paper in Science Translational Medicine describing the phenotype of MDMI and why we think they're a very strong model to study microglia in vitro. So with this model, we first set out to examine whether HSV1 infects MDMI and what the resulting phenotype is after infection. So to do so, we exposed our MDMI to HSV1 at 2 doses, a low MOI of 0.5 and a high MOI of 5. We incubated the cells with virus, washed them, and then measured readouts at an early acute time point at 6 hours and a subacute time point at 48 hours. And we measured infection by looking at the gene expression of viral genes in these cultures. And so we see that at both doses, at both time points, we see expression of HSV1 RNA in our cells. We also stained out the protein level for HSV1 using a polyclonal antibody against HSV1, and we do see that our cells are infected with HSV1. Now, this infection occurs in a dose-dependent way, such that regardless of time point, we see increased expression of viral genes at 6 hours, at a high dose, MOI of 5 versus a low dose, and it's also time-dependent, such that we see increased expression at time regardless of dose. Now, we really wanted to make sure that what we were seeing was due to true infection, so we exposed our, we actually heat inactivated our virus, and we exposed our cultures to heat inactivated virus, such that they were exposed to all the antigens in HSV1, but the viral particles wouldn't be able to actually infect the cells. And we see that in cells that are exposed to the heat inactivated virus, we see no transcription of our viral genes, suggesting that this is a true infection of MDMI. So now that we established our infection paradigm, we next wanted to better understand, well, how do these microglia that are infected with virus, what's their phenotype as a result? So to do so, we looked at the gene expression in our MDMI following viral infection with a panel of 100 pre-selected genes, including interferon response genes, cytokine genes, AD-associated genes, as well as general innate immune response genes. And we unsurprisingly see that HSV1 infection promotes a interferon response transcriptional signature. And this really is at the later time point, both with a low and high dose. So we see increased transcription of interferon response genes, including MX1, MX2, and ISG15. And we can concomitantly see a decreased expression of lipid regulatory genes. Again, really at the late time point, both at a low and high dose. So we see decreased transcription of genes such as NPC1 and FABP3. Now that we had started to understand the transcriptional response of MDMI to HSV1 infection, we wanted to look at other functional outcomes. So we next examined the effect of HSV-1 infection on phagocytosis of amyloid beta. So we exposed our infected cells to fluorescently tagged A beta and measured their phagocytosis. And we actually see that HSV-1 infection leads to increased phagocytosis of A beta. We were also really interested to understand the effect of infection on bioenergetics of our MDMI. So we performed seahorse on our infected cells to look at their cellular respiration. And really interestingly, we see that at baseline, cells that have been infected with MDMI have no change in their oxygen consumption rate compared to uninfected cells from the same individuals. But when we decouple their oxygen consumption from their ATP production, we see that their maximal respirations actually decreased as well as their spare reserve capacity, suggesting that these cells are less metabolically flexible and less able to increase or respond to increased metabolic demands associated with future challenges. So in this way, we've outlined the functional outcomes associated with HSV-1 infection in MDMI, as well as their transcriptional signatures. And we've really shown how infection really influences their phenotype. So our next question was, how do AD genetics modulate this infection? And we really focused on two different AD-associated genes, ApoE and PILAR-A. Ruth just beautifully outlined the association of ApoE4 and HSV-1 with AD risk. And PILAR-A actually encodes a sialic acid binding receptor that binds glycoprotein B of HSV-1 and is the receptor for viral entry of HSV-1 into the cells. So using the infection paradigm that I just described, we next examined infection in a genotype-dependent manner. So beginning with ApoE, we infected our cells based on ApoE genotype, and we find that at baseline, there is no difference in HSV-1 gene expression across cells of various ApoE backgrounds. But when we stratify by race, we actually see that in white and Asian individuals, that MDMI infected with HSV-1 actually have increased replication and increased load of viral gene expression in E4 carriers compared to E2 carriers. But we don't see the same trend in individuals of Black and Latino descent. And we next wanted to examine the effect of PILAR-A on HSV-1 infection of MDMI. And we see that PILAR-A genotype does not influence the viral load in our MDMI. However, really recently, beautiful work has shown the impact of this PILAR-A SNP on bioenergetics. So we were really interested to see how this PILAR-A SNP alters post-infectious bioenergetics in our cells. And while this is preliminary data, we actually see beautifully that while individuals carrying the AA protective PILAR-A variant are able to maintain their cellular respiration and their metabolic flexibility, MDMI from GG individuals following HSV-1 infection pretty much have just totally demolished oxygen consumption and are very much impaired metabolically following infection. So in this way, we've shown that AD genetics do influence the MDMI infection by HSV-1, both the viral load itself, as well as the cellular response to infection in the case of PILAR-A. Now, as Ruth so beautifully pointed out, most people are infected with HSV-1 early on in life. And Alzheimer's disease is a disease of aging, and something that's very important for us to consider as we're thinking about pathogens and their influence on developing AD is how exposure to an immune stimulus may change our cells over time. So something that I was very interested in understanding is how does an immune stimulus ultimately change microglial phenotypes longitudinally? And I turned to a phenomenon that's been well described in myeloid cells in the periphery called innate immune memory or trained immunity, such that exposure to a stimulus leads to chromatin remodeling. And cells are now, due to totally changes in their open chromatin, are poised to respond to a future stimulus in a different way. So this can be such that cells see an initial stimulus, remodel their chromatin based on that exposure, and are now poised to have an enhanced response to a future stimulus, or can have a suppressed response to a future stimulus, depending on how that chromatin remodeling shifted their ability to mount an immune response. So with that in mind, I was very curious as to what happens when microglia are exposed to HSV1 as an initial stimulus. Does it alter, like fundamentally change these cells at the chromatin level, and how does that change their ability to respond to future stimuli that they may encounter in the brain over time? So in order to examine that, I infected our MDMI with HSV1. I gave them a washout and rest period of 7 days. And then I performed ATAC sequencing to understand whether these cells had fundamentally remodeled at the chromatin level. And indeed, we found that compared to uninfected cells from the same individuals, that there were dramatic shifts in the chromatin structure of MDMI that had been infected by HSV1. And here I've shown we identified transcription factors that bind to these putative transcription factors that would bind to the sites of differentially open chromatin that we see following HSV1 infection. And these may be kind of mediating the functional outcome of how these cells are able to mount an immune response to future stimuli. So with that in mind, our next question is, well, how do these cells then respond to future stimuli? So I repeated this paradigm where I infected our MDMI with HSV1, and I gave them a washout and rest period of 7 days. And then I stimulated these cells with LPS to see how they were able to mount an immune response to this inflammatory stimulus. So we see that in cells that have never been treated with HSV1 or LPS, just the MDMA baseline, we see no transcription of inflammatory genes like TNF or IL-1 beta. When we treat cells only with LPS, we see this beautiful canonical increase, induction of IL-1 beta and TNF. Now to see like the baseline of cells that had been treated with HSV1 1 week prior, we see that these cells don't express pro-inflammatory markers. But when we look at cells that had been treated with LPS but had been exposed to HSV1 1 week prior, we see that these cells are unable to mount this pro-inflammatory canonical response to LPS. They have a fundamentally suppressed response. We also see this in genes such as STAT2, which is like a very important gene in the interferon kind of response. But very interestingly, while we see suppression in kind of these response genes that I've described, we also see like an enhanced or a hyperpolarized response in different pathways, such as in like the lipid regulatory pathway. In genes such as the fourth box, PLC-DX1, we see while LPS decreases its expression, concomitant HSV1 exposure, then LPS decreases it even further. So we see like a suppressed response to pro-inflammatory response, but we also see, an enhanced response in other domains. Now we wanted to better understand the functional outcome of this innate immune memory in a few other ways. So we, again, repeated our A-beta uptake assay, and we saw that in cells that had been exposed to HSV1 7 days prior, we see reduced A-beta phagocytosis following LPS stimulation compared to cells that had only been stimulated with LPS, again suggesting this kind of like suppressed response and hypoability to be able to respond. We also looked at bioenergetics in these cells, and we find that again, 7 days post-exposure following this remodeling, we see reduced metabolic fitness following this kind of rewiring. Again, further showing that these cells are not able to functionally respond in the way that microglia are at baseline. So in summary, our work shows that HSV1 does infect microglia-like cells. And the result of that infection is that it activates interferon response and increases phagocytosis and alters bioenergetics of these cells in the acute phase. and that this infection is regulated by AD-associated risk variants, including ApoE and PILRA. But that this exposure, this immune exposure, ultimately rewires the chromatin structure of these cells, resulting in a sustained suppressed response. And ultimately, the goal of this work will be to understand how this innate immune memory and this kind of immune learning that microglia undergo is mediated by AD genetic risk variants, and how all these factors together can push us to understand microglial dysfunction that may ultimately contribute to AD pathogenesis. So with that, I'd like to thank everybody who's contributed to this work. We've had several collaborators. Those in our lab, Mato Yerink and Kristen Tamucci, have really helped with the HSV1 functional studies that I've showed, as well as Jason Mares in the Menin lab, who's helped with the ATAC sequencing analysis, and Jackie Eisman, who helped us with the transcriptional analysis. So, and thank you so much again to Nikki for organizing this day.
Right. No, there's no time. Yeah, there's no time. Yeah, if you have questions, you're later available to take them. I'm waiting for Professor Edward Breitschwerdt and Janice Bush from North Carolina State University. He's a professor of infectious disease, and he will talk about Bartonella and neurodegenerative diseases related to Alzheimer. I don't know how to move these things around. Yeah, I can.
I want to thank Dr. Balin and Nikki for the invitation to present. Wait a minute. Try this down here. That'll work better. Can you hear me? Yeah. All right. Our talk is Bartonella henselae, neurobartonellosis, and Alzheimer's disease. Is there a link? Janice Bush is a PhD student in my laboratory. In the context of introduction, I had a previous PhD student generate this particular slide. And I think it's pertinent to what we've already heard this morning and more of what we're going to hear. And that's that bacteria, which has been the primary focus of our research, have been on this planet for 3.5 billion years. They have co-evolved with ticks, insects, primitive humans. And it was just over 100 years ago that we actually determined that ticks in Texas could transmit Bartonella bovis to cattle as the first vector-transmitted infectious disease. I think the other concept, I'm a veterinary internist. I still see sick cats and dogs part of my time. And this concept I like very much as a clinician, and that's the fact that there is a pathobiome that can influence the microbiome, and that ultimately influences what goes on in the host. And my hope as a clinician is that there's a finite number of organisms that constitute the pathobiome, because if not, this is far too complicated for me to deal with on a clinical basis. And trying to think of something that was useful that we've done that I might be able to share with the audience. The question is, in the context of an Alzheimer pathobiome initiative, is there utility in looking for the presence of a specific pathogen or combination of pathogens at different life stages? And as I reflect on our research, we've actually done that somewhat indirectly. So in 2010, we published a paper in which we were able to get tissues from a little girl that died of 8 days of age. And 10 years later, we were testing the parents and the son that has survived. And in working with a human pathologist in New York, we were able to demonstrate that both the mother, the son, and the child were infected with Bartonella vinsoniac burgdorferi and Bartonella hensley. So transplacental transmission wasn't proven, but at least perinatal transmission certainly did occur. This is a situation that we've been working on since 2022. It's a young girl seen at Sick Kids up in Canada, and she had a cat scratch by a feral cat when she was 2 years old. She then had some unusual, and I'll show you pictures of both on the next slide, insect bite when in 2018. Within 2 weeks of that second hit, which I'll refer to it, she started seizuring. Sequential MRIs suggested she has a disease called Rasmussen's encephalitis, which is a unilateral hemispheric encephalitis where the treatment is to essentially cut out portions of the cortex on that side of the brain. And in June of 2022, because of the history of cat scratch, we isolated Bartonella hensley from 2 independent brain cultures using 2 different medias, suggesting that for at least 6 years, she had viable Bartonella in her brain. And this is the picture of the scratches that this child experienced, and that's the bite on the hand. We've subsequently demonstrated that most likely that was a tick bite, and the organism that was transmitted was Babesia otocolii. So I think the second hit was the one that allowed the Bartonella hensley that was insisted to perhaps induce the seizures. We'll now go to a 14-year-old boy with pediatric onset acute neuropsychiatric syndrome. And this was published in Central Nervous System Disease in 2019. And this was a rather, as happens so often with these children, sad story in which this boy had become suicidal, homicidal. You can see he failed 18 months, which I'll show you on the next slide, of various neuropsychiatric medications because he had these lesions, which when Lyme literate physicians suggested to me that these lesions were prototypical for Bartonella, I shrugged and didn't believe it, but I do believe they're not necessarily prototypical, but they certainly do occur in some people that are infected with Bartonella. And we call them Bartonella-associated cutaneous lesions. They've been referred to as stria or tracts, but we certainly need some human dermatologists to figure out what the heck's going on in regard to these lesions. I think the importance of this slide is you can see the number of physicians, the number of specialists, and the number of therapies that this young man experienced. And the, I guess, importance for me is nearly $500,000 had been spent at the point that we documented that he had Bartonella hensley in his blood. You can also see from this particular figure that the initial therapies through 2015-16 up through October were a spectrum of neuropsychiatric drugs. And as I mentioned, he was treated for autoimmune or immune-mediated encephalitis. And none of those treatments essentially helped him until they started treating him with antimicrobial drugs. At that point, and within 6 months, he was off all antipsychotic medications. And I can tell you at this point, he is now cleared, has been off all medications for 2 years, and is a pre-med student at The Ohio State University. We'll now go to middle-aged individuals, which our research essentially with Bartonella went cats, dogs, horses, cows, marine mammals, and then ultimately sick veterinarians and veterinary technicians. So it's very biased towards my profession. And which you can see here, and I'll emphasize the first case, which was not a veterinarian, at 21 years old, this young woman was diagnosed by a dermatologist as having classical cat scratch disease. Within a year, she started with minor motor seizures, then progressed to generalized seizures. And what you can see, and this was a collaboration with Chris Woods, a infectious disease physician at Duke University Medical Center, that we could consistently document that she was infected with Bartonella henselae, we could also strain type that down to the Houston 1 strain type that was initially isolated from an AIDS patient in Houston's. I think the importance here is we were able to isolate Bartonella from the brain of the child that I mentioned, and we were also able to document in culture that there seemingly was viable Bartonella in the cerebrospinal fluid of this woman literally 2.5 years after she was diagnosed with cat scratch disease. And at this point, we were collaborating with William Nicholson at CDC, who did the serology. And serology seems to be somewhat helpful in following patients, because I think if we can truly eliminate this organism, then patients will tend to go seronegative. And that's documented in most of what we've tried to publish. So, these got left in. We're going to skip those in the interest of time, or my PhD student will shoot me. So more recently, our studies have focused on neuropsychiatric illnesses. We've been very fortunate to do 2 studies, one with Flavio Follick's group at UNC, and the other with Shannon Delaney and Brian Fallon at Columbia University. And if you look at the summary of these 2, you'll find that statistically, if a person has either psychoses in New York or a diagnosis of schizophrenia in North Carolina, we can amplify and sequence, I shouldn't say that, by digital PCR, we can document statistically that they have Bartonella DNA in there, some of whom we can actually document by sequencing what the Bartonella is. And this is the most recent study that was just accepted in, again, working with Flavio Follick's group at UNC. And we decided to look at individuals with mild cognitive impairment. And in the interest of time, I'll just I'll point out that this is the highest digital PCR result that we've gotten in any group of individuals that we've looked at to date. So it may be that as we get older, the ability to keep Bartonella suppressed and within endothelial cells and other places in which this bacteria hides as a niche is less effective, and we're more able to find it in circulation, but that certainly requires more work. Janice?
Thank you, Dr. Breitschwerdt. Can I take the remote from you?
Whoops, sorry.
Thank you, so I don't have to say adjust slide. So we were interested, other way. We were interested in what was the common ground between neuropsychiatric and neuroinflammatory or neurodegenerative disorders. And we have heard a lot today about this inflammatory pattern that exists. And this is evidenced by changes in cytokines. It's evidenced by abnormal immune activation within the brain. And also there's evidence of cellular damage, atrophy, apoptosis. So this is my biorender image of the blood-brain barrier. It might be a little bit devoid of some other cells, but we wanted to put it up to talk about the ways that things might be able to get across. So from the systemic side, we can certainly have bloodborne pathogens. So many people have talked about inflammatory cells, even platelets, antibodies, shifts in vascular permeability that will cause changes. And then from the brain side, we're going to have those microglia, I think my favorite cell now, that is reacting to various stimuli, also involved in extensive crosstalk between neurons, astrocytes, brain vascular pericytes, and the vascular endothelium itself. So chronic microglial activation and microglial activation in general does appear to be really a main instigator in some of these conditions and changes that we're seeing. And in fact, it's been shown that patients with Alzheimer's disease will develop psychiatric symptoms upon microglial activation, and as well, that glial activation may actually precede some of the abnormal protein aggregates that we see in various neurodegenerative disorders. So in this diagram here, we wanted to point out that the microglia have kind of this dual functionality. like any macrophage, of course, they're going to be responding to invading pathogens, fungi, viruses, bacteria, and whatnot, but also responding to cellular damage. And you can see the increase in inflammatory mediators associated with these things. On the flip side, they also are producing these homeostatic factors. I've got IL-10 up there and some of the type 1 interferons that are going to be involved in maybe helping to quench the inflammatory response. But especially with regards to IL-10, really important in neuronal glial crosstalk. Elevated IL-10 or IL-10 in general will allow the neurons to produce CX3-CL1. And this is important in signaling to the microglia that we need neuroprotection going on. So it can help to modulate the immune response. But what about persistent elevation of IL-10? We always think of IL-10 as kind of this canonical anti-inflammatory cytokine, and that should be a good thing. There's a lot of talk about inflammation. Maybe we should focus on anti-inflammation. But it might not be the best thing for us. And there are a lot of conditions that are associated with chronically elevated IL-10 levels, multiple autoimmune conditions, as well as neuropsychiatric issues, and even Alzheimer's disease potentially in some studies. So we're an infectious disease lab and excited to be here based on that. But as infectious disease people, I want to talk about our pathogen, which is Bartonella henselae. And part of what it does as an intracellular bacteria, it needs to create a hospitable environment for itself. It doesn't want to get killed. And one of the things that it does is it has a really ingenious way of hijacking cellular machinery in order to cause the host cell to bump up IL-10 production. So you can start to see that there might be sort of this chronic, or maybe it comes in waves, production of IL-10 that could be going on in an infected cell or an infected organism. So too much IL-10 or too persistent of IL-10 does appear to be involved with decreasing the amount of fractalkine or the CX3-CL1 that's expressed by neurons, and also diminishing its expression of its receptor on the microglia. So over time, this might cause neuronal degeneration, and you could see that you might have an inflammatory situation become propagated through cellular damage and then activation of damage-associated markers in the microglia. Elevated IL-10 has also been shown in a mouse model to diminish phagocytosis. And so we started to be just really interested in this. You know, we know our bacteria can cause elevated IL-10. Is that potentially the trigger? And it's not just necessarily a reaction to pre-existing inflammation. So we wanted to look at our bacteria in a human glial cell line. But first, we had some previous research that showed that Bartonella henselae was indeed able to infect feline microglia cells. So the cat is this bacteria's natural host. And in the electron photomicrographs there with the red arrows, we're seeing intracellular bacteria. And interestingly, in this study, it was shown that those bacteria were able to be cultured after infecting the cells for a period of up to 28 days. So it seemed as though these were living now in the feline microglia cell for up to 28 days. We looked at the HMC3 cell line, which is a human microglial line, and the image there, the immunocytochemistry image, is just from a 48-hour post-infection. But we have grown it successfully up to 11 days post-infection in that line. So we want to know what the microglia or how they're responding to this. And this is really preliminary data that I have right now. What we did was we did a phagocytosis assay comparing what the glia were doing after being infected, how were they phagocytizing, was it going up or down, compared to a different cell line, the DH82 monocytes, which are a canine monocyte and really proven ultimate phagocytosing cells. And you can see here that it does look like, compared to our baseline, the DHAD2 monocytes tend to bump up phagocytosis over the 48-hour infection period. And again, really preliminary, but it seems as though our glia are decreasing their phagocytosis. So kind of maybe going along with, are we seeing a shift in the ability? The data that we want to present today, unfortunately, won't be back until probably later this week. It's a day late and a dollar short, but the hope is that, we'll be able to look at the transcriptomic data associated with this as well. Will we see changes in IL-10s? We've got some of those studies going on as well. And we're thrilled to be here. We are veterinarian. To be among medical doctors and researchers is just amazing for us. So I want to thank our lab, who are wonderful to work with, our funding sources, the Cohen Foundation, and the collaborators in the research. And a special thanks to Dr. Balin for including us in this amazing journey, and to Nikki for setting it all up, and as well as the grant with NIH. And we can't not thank Bartonella because it would get mad.
We have time for questions? No time for questions. I'm sorry. So, without further ado, we go and invite Dr. Shemesh and Vanesa Hyde from Jerusalem University and University of Pittsburgh, and they will give us a talk all about spatial imaging and expansion microscopy, looking at the infectious agents in the brain. So.
Can't hear?
No, talk nice.
Closer. Closer. Sorry. Hi, everyone. My name is Or Shemesh. I'm a assistant professor in the Hebrew University of Jerusalem and the University of Pittsburgh, where most of this was performed. I'd like to thank Nikki and Brian for arranging this amazing conference, showing that a lot of people are in the field, and the field is advancing and changing. So let's do it. I'll drink a lot of coffee because I'm a bit groggy. I'm jet lagged, and also yesterday Brian Corey and Garth Ehrlich gave me a lot to drink, and I'm going to complain later. I'm a letter writer. Let's begin. So these are my grandparents from a mother's side, Abraham Sitton and Rebecca Sitton. He was diagnosed, my grandpa was diagnosed in 2000, and my grandma was diagnosed in 2014. And the treatment was similar. The prognosis, as you might guess, was similar. And when my grandpa was diagnosed, I went to the doctor, the MD, and I asked at the time, before I, even my bachelor's degree, what's Alzheimer's? He said, A, it's a disease where you have inflammation in the brain. B, prognosis is very bad. And C, we won't have any cure within the next 100 years. So I delved into neuroscience and focused on on neurotechnology, creating tools to study the brain rather than just doing basic science. I did my postdoc in MIT, and by the end of it, I still didn't know what causes Alzheimer's disease. But this is what we know of the pathophysiology of Alzheimer's. We have, after several decades of research, we know that there's a decrease in synaptic density, an increase in A beta deposition, an increase in the activation of astroglia, microglia, and astrocytes, and we have an increase in tau pathology, as you know. And over the years, as I was studying, I did my PhD at the Hebrew U on Alzheimer's disease. There are a lot of papers that I came across that are really, I call them the red flags, like there's the fact that, you know, there's inflammation in the brain, and a very common cause of inflammation, as you know, are pathogens. The fact that the disease is progressive, you have a frontotemporal distribution, and pathogens often form frontotemporal distribution. This is also what we found later on in Alzheimer's brains. And I give a quote here from a paper because I had a lot of backlash when I didn't. A subject whose spouse experienced incident dementia onset had a 6 times greater risk for incident dementia, a subject whose spouses were dementia-free, which is a big clue that this could be infectious and that we shouldn't get married. And I'm sorry. And age, of course, is a risk factor, alluding to the cumulative damage. And very interesting, multiple comorbidities. We have 184 patients, only 30% of them had only AD. The others had AD and alpha-synuclein pathology, AD and TDP43, another interesting protein pathology. So if we have these discrete pathologies, maybe we have discrete causes, maybe discrete pathogens. So, as you know, the pathogen hypothesis of AD, of Alzheimer's disease, and PD, and ALS, and whatnot, proposes that either viruses, bacteria, fungi, prions, some of all of the above play a causative role in development of the disorder, of the syndrome. And I'm going to talk today about HSV1, herpes simplex virus 1. I just want to be very clear that as much as I think that HSV is a huge contributor to the disease, we're not proponents of the idea that it's only HSV. We see and we do studies showing that there's a vast microbiome for AD, which is different from the microbiome of Parkinson's, which is different from the microbiome of other diseases. But what I'm going to show you is that technology really can tell us new things about pathogen and about a disease. And as you know, Ruth Itzhaki is the pioneer of these studies. And she and later others have detected that in patients of Alzheimer's and also in non-patients, there are a lot of HSV1. And ever since the 70s, you see, in the 80s, there were case studies showing that people with Alzheimer's and also non-patients had HSV, serological samples, in situ hybridization, a lot of sequencing. And after several decades of research, here's what we know. HSV1, herpes simplex virus 1, was found in the brains of most elderly people. However, we can't see active infection, that much active infection. Where are the variants? Where are the infective particles? 2, there's productive AV infections that are linked to the AD pathology, but AD patients rarely die of encephalitis. So why don't we have encephalitis? This is rarely the cause of death. So what we hypothesized is that it's subproductive infection that leads to the pathologies of AD. There's a little bit of expression all the time. And at a certain point, we cross a tipping point and we have some of the manifestation of Alzheimer's disease. So we started looking into sequencing and finding proteins, the proteome and the genome of HSV1 in AD patients. We had some brains coming in. This is just an example. We have more than 10 brains of advanced AD versus non-AD patients, and we sequenced them, and we found HSV genes in all of them. So nothing unique there. We didn't find a difference between the AD to the non-AD, to our controls. Then we said, okay, this was done with collaboration of Jose Lugo Martinez from CMU. I reached out to my friend Daniel Barr. I said, let's play with the mass spec. Here's some of the analysis. We found 12 genes of HSV1, both in the patients, no, 12 proteins, sorry, patients and non-patients. Again, what's the difference between the patients and non-patients? So we said, okay, let's try to target specific proteins of HSV1. And we did some Western blotting of patients. These are HeLa cells, non-AD, mild AD, and advanced AD. We looked for GB, that's a late protein, and for ICP27 and 4, immediate early proteins. Everything was positive, always. I know that we've been taught that the positivity in the population for HSV1 is between 63, 65 to 90 in different studies. I haven't seen a single brain without proteins of HSV1. So over the last 5 years. So again, we were kind of stuck because everything was positive. So how can we distinguish the patients from the non-patients? And then what we wanted to do, by the way, this is Pablo Valdez. He worked with me in MIT where I did my postdoc. And he invented a technology in Ed Boyden's group to look at the brain and at biological samples at superior resolution, which is called expansion microscopy. Basically, we wanted to image viruses and other pathogens. However, they're very small. HSVs are around 150 nanometers across to 200 nanometers. And also the antibodies, we used antibodies, they have a non-negligible size. They're big. So basically what we wanted to do was to do this, expand. Expansion microscopy is not a super resolution technique, it's a tissue manipulation method. You take the tissue, you expand it, then you go with the same run-of-the-mill microscope and you image things at 20 to 40 nanometer resolution. So that's what we wanted to do. And we do it with multiple pathogens and several diseases. We expand the human brain, we label, and then we image. So just to show you what we do, let's say you want to image these microtubules here. We use fixed samples, FFPEs, and we add to them an anchoring molecule. The anchoring molecule binds to all the proteins in the cells, or to most of the proteins in the cells, and then we infuse a diaper polymer that also binds to the anchoring molecule. And now all our proteins are held by the anchor. Then we cook it at high temperature, 120 degrees, with SDS and with beta-mercapto, with very strong denaturing and reducing agents. And all that's left is the gel. We add water, it expands, then you can label. You have decrowded the epitopes. Let's see if it's any good for AD. I just want to show you the same tissue, not the same field of view. I was lazy at the time. The same tissue from an AD patients, the same AD patients, before expansion, after expansion. These are the A-beta oligomers, one of the toxic moieties of Alzheimer's disease. So here we identified something like 56 puncta, and here a 10-fold increase in the number of these positive puncta. You can see things you couldn't see before. Now, another thing we did, we looked at the same brain and compared low Alzheimer's disease patients before expansion, after expansion, and advanced AD patients before expansion and after expansion. In green, we labeled neurofilaments, neurons. In red, this is magenta, I'm colorblind, we labeled p-tau, hyperphosphorylated tau. And basically what we saw is that after expansion, you don't see much p-tau, but before expansion, In the same patients, it's positive. This is artifactual. Everything you see here is artifactual. This is called lipofuscin, which is basically oxidized DNA and oxidized proteins and whatnot that we have, and we have in the elderly brains, and they're autofluorescent. So look how before expansion microscopy, how correlated the signals are between the neurofilaments and p-tau. And look how much p-tau we have pre-expansion, and then it, boom, disappears post-expansion. So basically, we eliminate lipofuscin. We have a far superior resolution, as you can see here, and we don't have artifactual correlations or colocalizations between proteins when we do expansion microscopy. And now I'll move the stage I'll give the stage to Vanesa, who actually did these experiments.
All right, so the most important thing is to address our cohort. So our data is super unique because we use expansion microscopy, which enables us to use our human brain samples. And so our human cohorts were from the University of Pittsburgh ADRCs and the University of California, San Diego. And so for this, we use 3 brain regions: the entorhinal cortex, the hippocampus, and the cerebellum. And our diagnoses were non-AD, mild AD, and advanced AD. So no AD would be backstage 0 through 2, mild AD would be 3 and 4, and advanced AD would be 5 and 6. So the first step in this was our target was HSV1, as we have addressed a little bit earlier here. And so our first target was to actually see if we could identify and see HSV1 in human brains. So on the left column, you will have the entorhinal cortex, and on the right column is the hippocampus. And on the top row is no AD, middle is mild AD, and bottom is advanced AD. And so we have neurons in red, microglia in blue, and HSV1 ICP27 in green. So we selected ICP27 based on its very vital role in both viral replication, along with its hand in a lot of other viral functions that make HSV what it is. So as you can see in the top row with no AD, there's very light detection of the ICP27. You can still see a few particles, but it's nothing too dramatic. As you move down to advanced AD, you can definitely see a much larger green stain signaling the ICP27. So visually, definitely a change as you move into the advanced stages. So to quantify this, we did something that we call the area of expression. So basically, we generated a threshold of what we would determine as positive pixels, and then calculated how much of the image was actually covered with these pixels to kind of assess how many ICP27 particles we're seeing. So as you can see with our top graph, we have the ICP27 analysis, in which we have significant increase from the no AD condition to the advanced AD condition. These tend to be 2 to 3 times more pixel detection compared to the controls. And so we also looked for ICP4, GB, and BP16. Although there were no significant differences between them and no trends we could really identify, they were technically detected. So we wanted to make note of that as well. And so after we could see the ICP27, we wanted to know where it was and what it was doing. And so we noticed that with our cell types, we were seeing a shift in them. So our first analysis was basically to assess where the ICP27 was located. So in the bottom left-hand corner, you have cocalization scatter plots from a single human brain image, in which we can see as you move to advanced AD that there is a shift into the microglia. We then did this similar analysis with the Mander's overlap coefficient of our images for our entire brain cohort, and you can see there is a significant decrease in the amount of cocalization between ICP27 and neurons, and we see a significant increase in the amount of ICP27 cocalizing with microglia. I would like to make a note that the overlap percentage for microglia is relatively low in comparison, just because there's less of a microglia stain in comparison to the neurons. And so we also wanted to show this as a step-by-step. So the top panels of images are actually the serial images themselves. So you can see in teal the ICP27 with the microglia.
And so from here, what is Alzheimer's disease research without looking at some of our classic pathologies? So to show our technology works, I stained a lot of our conical AD sites. So we have 88, which is 202, 205, 199, 202, 205, 231, 214, and 396. For the rest of these experiments, I selected 396 just based on its very bright stain and its clear depiction in the human brain. So from here, we were looking to see if ICP27 co-localized with our human brains. Again, the orientation is very similar with the no AD on the top and the advanced AD on the bottom. We have ICP27 in green and p-tau 396 in red, and then the large images have the overlap, so the yellow is their co-localization between them. So as you can see from the top, there is basically no ICP27 stain, and as expected, basically no p-tau. And as we move into our advanced stages of AD, we see this very beautiful co-localization in the human brain between ICP27 and p-tau. And so again, we ran a co-localization analysis, and we have a significant increase in both the entorhinal cortex and the hippocampus from no AD to advanced AD in this stage. So you can see the co-localization in the images above. So we have our controls so that we know that our antibodies are actually labeling what they're supposed to. So this is our negative control, showing that when there's no infection, there is no stain. And we also have our positive control, where we did Western blot in infected cultures and transfected cultures, showing that our antibodies are labeling exactly what we're supposed to. And so we couldn't forget amyloid beta. And so we did amyloid beta plaques and amyloid beta oligomers. And in this, we do not see the co-localization that we saw with the tau. And so we ran analysis comparing them, and you can see the levels for the plaques and oligomers is way below those of the p-tau. And so we moved into modeling this, because up until this point, it's basically all observation-based. And so we collaborated with Leonardo D'Aiuto at UPMC Hospital, and he forms cultures from iPSC neurons. And so this shows we did Western blots with an infected culture of a 2D culture. And so we have increased tau phosphorylation following HSV1 infection. So moving into, he developed 3D brain organoids for us to use. And so he ran the experiments for these. So his conditions focused on an uninfected control, a acute infection of 3 days, an acute infection of 5 days, a latent infection, which was basically antiviral treatment, and then a reactivation condition, which mimics what we believe might be happening in the Alzheimer's disease brain. So we targeted 2 different p-tau sites. And just as a quick summary, as we're short on time here, we noticed that the p-tau phosphorylation increased with infection, as you can see by our significance and our graphs there. But basically, HSV1 infection causes tau phosphorylation. Many of you have known this or shown it yourself. So we also focused on the actual co-localization between them within the brain organoids. And so you have day 3 infection, where you have a pretty nice overlap between the p-tau and the ICP27. And then moving into day 5, we have an overwhelming presence of the p-tau itself, in which we believe that the p-tau is starting to overwhelm the infection itself. And then the latent infection shows that there's no tau phosphorylation when treated with antivirals, and the reactivation condition has a similar resurgence that we see in the day 3 infection. And so some of his earlier work complements this nicely, although it has been shown that 2D cultures have a development of amyloid beta with HSV infection. When he ran a 3D brain model, he actually found that the amyloid beta was in adjacent cells, not positively infected cells, as he stained with ICP26 or ICP4. And so as a summary, we use expansion microscopy to show ICP27 in the brain. We notice ICP27 shifts to microglia in advanced stages of AD, and that the ICP27 from HSV1 co-localizes with p-tau. And then when modeling, we know that HSV1 leads to an increase in tau phosphorylation and co-localizes with the neurons. And so what is next? So our continued research focuses on searching for the biological purpose of tau phosphorylation. To do this, we are using a primary neuronal culture in which we have tested out and we see the exact same phosphorylation trend in them. So they should be ideal for a quick, easy model. And so for more data for this, you can visit me on Wednesday at my poster AIC if you are in attendance. And I would like to thank our team back home in Pittsburgh.
You have just 5 minutes per question. Actually, I have a question for you from the online audience. And someone was asking whether this expansion microscopy technique does not mask the epitope of phospho-tau in low AD subjects. The dope of phospho-tau, and so that you don't get strong phospho-tau in low AD.
This one.
Yeah.
The question was whether it masks the phospho-tau in
The
whether
technique was-
expansion.
Yeah.
We don't see any indication of epitope masking. On the contrary. So expansion microscopy, the one thing we had to test repeatedly was the isotropy, whether we expand it in 3D in the same to the same extent. So there is some error there. So that's what I can tell you. It really depends on how thin your sample is. In terms of showing or masking epitopes, what we see is that we do have, even in our data, we do have the phospho-tau in non-patients, in non-AD, but it's to a much lower extent compared to the patients.
Okay, thank you. Then, Garth.
Hello, can you hear? So you're saying that 100% of the specimens you looked at were HSV positive. Were they also by DNA or protein? I forget where you maybe it's pretty. Are they also seropositive? Are they all seropositive as well? Or do you see this amongst seronegatives?
We don't have the seropositivity or negativity of these patients. We'd love to see, because it would tell us how telling is the seropositivity, how telling is the blood samples, how telling is the CSF, how representative it is of what's happening in the brain or not. But we don't have it. All DNA, all metagenomic sequencing came back positive, and all mass spec came back positive, and all expansion microscopy came back positive. That I can tell you, but not, unfortunately.
I still have one question from the audience to Professor Breitschwerdt and his colleague Bush. They're asking whether Bartonella can infect gums. Yeah. Yeah, better. Yeah.
I was just responding that on text to Nikki. The answer to that question is we know you can find Bartonella quintana in the dental pulp of Napoleon soldiers. We have amplified multiple Bartonella species out of the saliva of dogs. Other researchers, many have amplified and sequenced it out of cats. Cats get a very unusual and very devastating disease called gingivitis stomatitis, which the current treatment is pull all the teeth out. And I think that, like Alzheimer's disease, that condition is probably very complex and multifactorial, but clearly you can find plenty of Bartonella in that particular lesion.
Thank you. There was one more question back there, close to you.
Very interesting presentation. So I have several questions, but I would like to know more about the technical part of tissue expansion. But I have a couple of questions about the patients that you used to run the tests. I wonder what were the ages of the patients, the sexes, and the BMIs?
So the ages for all three, low AD, no AD, or no AD, mild AD, and advanced AD, there was no significant difference between them. They ranged from 70 to 95. And then we did not divide by sex, so there was both male and females in our group. And in terms of PMI, most of them were under 10 hours.
Under 10? The body mass index was,
Oh, you mean BMI. So I thought you said
BMI.
BMI. BMI, we do not have that data.
yeah, I did. Okay. And do you know what was the criteria to diagnose these patients with Alzheimer's disease and not dementia?
It was their pathogenic report from the ADRCs, so the typical tau load and amyloid beta load and.
Okay.
Okay, I think we're going to have to stop with the questions right now because lunch is waiting. Lunch will be up on the mezzanine as we go out of here. If you make a right and walk up the steps, it's up on the mezzanine. We're running a little bit behind schedule, so we might have to go with lunch a little bit shorter than 50 minutes, maybe 40 minutes or so, and then we'll try to reconvene right after lunch, okay? Thank you all.